Physiology of the blood. Blood is made of 55% plasma, 45% erythrocytes, and less than 1% platelets and leukocytes. The three major cell types in the blood are erythrocytes, leukocytes, and platelets. Erythrocytes are red blood cells. Their main function is oxygen transport. Leukocytes are white blood cells, and their main function is immunological defense. Platelets, homeostasis. Plasma is 90% water, some electrolytes, nutrients, wastes, gases, and hormones, and plasma proteins. Plasma proteins include albumins, fibrinogen, and globulins. These plasma proteins are made in the liver. Albumin is very abundant and contributes to colloid osmotic pressure. Fibrinogen has a clotting factor, and globulins are used as transport materials, where the gamma immunoglobulin makes antibodies and is made in the lymphocytes. All the other plasma proteins are made in the liver. Hemoglobin. Bonds between hemoglobin and oxygen are fairly weak. This is for quick attachment and detachment to and from the hemoglobin. So oxygen binds to hemoglobin in the blood vessels in the lungs, and oxygen leaves hemoglobin in the blood vessels of the body tissues. Hemoglobin also binds to carbon dioxide, carbon monoxide, nitroxide, and it buffers acids to minimize changes in blood pH. The acidic H+ part of the ionized carbonic acid. What is an erythrocyte? An erythrocyte is a red blood cell that is anucleate. It has no organelles or ribosomes. It has a biconcave shape for a large surface area. It's thin, it's flexible for efficient diffusion, and it carries oxygen or carbon dioxide. Each erythrocyte has around 280 million molecules of hemoglobin, and each hemoglobin has 4 iron components and can bind 4 oxygen molecules. The process of haemopoiesis. What is the process of haemopoiesis? Well, haemopoiesis is blood cell production via progressive differentiation. That is, one undifferentiated pluripotent stem cell gradually acquires specific traits of end cells. So, the undifferentiated pluripotent stem cell can differentiate into myeloid stem cell or lymphoid stem cell. And if you go with lymphoid, you get lymphocytes, such as the B lymphocyte and the T lymphocyte. The myeloid stem cell is a lot more complicated. It differentiates into megakaryocytes, which form platelets, granulocyte precursors, which forms granulites, including basophils, eosinophils, and neutrophils, erythrocyte precursors, which of course form erythrocytes, and monocyte precursors, which form monocytes. So erythrocyte enzymes, they're the enzymes involved with erythrocytes. So there are glycolytic enzymes, and these are needed to make energy, which fuel the active transport mechanisms involved in regulating ion concentrations in the cell. And these enzymes, as their name suggests, rely on glycolysis for ATP formation. The second big enzyme to know is the carbonic anhydrase enzyme. This is needed for carbon dioxide transport. And what it does is it catalyzes the reaction that converts metabolically made carbon dioxide into the bicarbonate ion, HC3O-. And the bicarbonate ion, HC3O-, is the primary form in which CO2 is transported in the blood. So talking more about red blood cells and erythropoiesis, and erythropoiesis is red blood cell synthesis. How does that happen? It starts off as a hemocytoblast, which is a stem cell. This becomes the proerythroblast, and is stimulated by erythropoietin, EPO. So hemocytoblast to proerythroblast to erythroblast to normoblast, where the nucleus is expelled, to reticulocyte and erythrocytes. So that's 4 blasts and 2 cytes. Hemocytoblast, proerythroblast, erythroblast, normoblast, reticulocyte, erythrocyte. One more time. Hemocytoblast, proerythroblast, erythroblast, normoblast, reticulocyte, erythrocyte. Approximately 1 trillion red blood cells are made every day, and reticulocytes make up around 1% of all red blood cells. So, EPO, erythropoietin, what is it? Well, it's made by the kidneys, and what it does is it stimulates erythropoiesis, red blood cell synthesis. So, how does it work? Well, kidneys detect a reduced oxygen carrying capacity of the blood, and when less oxygen is delivered to the kidneys, they secrete EPO, erythropoietin, into the blood. Erythropoietin, EPO, then stimulates erythropoiesis in the bone marrow, and extra circulating erythrocytes increase oxygen carrying capacity of the blood. So, of course, when that happens, it relieves the initial stimulus that triggered the EPO secretion, and the whole situation is resolved. Increased erythropoietin is produced at higher altitudes naturally, and synthetic erythropoietin that's used clinically is called epogen procrit. So what is the lifespan of the erythrocyte? Typically, erythrocytes are formed in the red marrow. They circulate in the bloodstream for around 120 days. They are eventually phagocytosed in the liver or the spleen, and then its components are recycled. So the heme component is converted to biliverdin and then bilirubin and is secreted in bile. The iron component is transported in blood via transferrin and stored by protein ferritin in the liver. Membrane proteins and other globin proteins and such like that are broken down into amino acids, and some of them themselves are actually reused to make new red blood cells or other little proteiny things. So, in summary, erythrocytes survive 120 days in circulation. Old erythrocytes are removed by macrophages and in the spleen or the bone marrow, and iron is recycled back into haemoglobin production. So when subnormal oxygen carrying capacity of the blood occurs, we call it anemia. And some possible causes of anemia is a reduced hemoglobin content of the red blood cells, such as having an iron deficiency, or a reduced red blood cell number. So cell production is maybe reduced, or there's just extra cell losses incurred. So different types of anemia are nutritional anemia, where there's an iron deficiency, pernicious anemia, where there is an inability to absorb vitamin B12, aplastic anemia, where there's a failure of bone marrow to make enough red blood cells, renal anemia, which is impaired erythropoietin synthesis in the kidneys, hemorrhagic anemia, extreme blood loss, and hemolytic anemia, where red blood cells rupture maybe due to malaria or sickle cell disease. Another blood condition is polycthemia. So primary polycthemia is a tumour-like condition of the bone marrow where erythropoiesis is uncontrollable. And secondary polycthemia is where erythropoietin induces a training scheme during cases of prolonged reduced oxygen delivery, such as mountaineers at high altitudes, or people that are living with impaired oxygen delivery due to lung or heart disease. So what exactly is polycthemia? Well, it's just too many red blood cells and elevated hematocrit and increased blood viscosity. The blood is just too thick with too many red blood cells. Normal hematocrit levels in regular healthy human, 45%. When somebody has polycthemia, hematocrit levels reach 70%. Quite a big difference. Next thing I'm going to talk about is platelets. And platelets are formerly known as thrombocytes. The cell formerly known as the thrombocyte. Characteristics of platelets. They lack nucleus. They are fragments of the megakaryocytes. They're massive blood clots. What they do, they release serotonin to vasoconstrict and reduce blood flow to a clot area. They secrete growth factors to maintain integrity of the blood vessel wall. and they survive for up to 5 to 9 days and are eventually removed by macrophages. Thrombopoietin. What is thrombopoietin? Well, it's a hormone that is made by the liver, which increases the number of megakaryocytes and thus increases platelet production. Homeostasis is one of the most important defenses we have as humans against pathogens, and one of the most important features of thrombocytes as well. One of the most important functions, excuse me. So what is hemostasis? It is the process of keeping blood within a damaged blood vessel. How does it work? 2 main steps. 1, formation of a platelet plug. 2, blood coagulation, clotting. So how does that work? What's the clotting all about? Well, that occurs through a blood clotting cascade through 2 pathways. One is extrinsic, short and fast, and the other is intrinsic, and it just needs a lot of upstream factors, and I'll get to that in a bit. But let's just talk about the formation of the platelet plug first, and how does that work? Well, what happens is that platelets, they aggregate on contact with the exposed collagen in the damaged wall of the vessel, and then what happens is the platelets increase ADP in the area, release ADP in the area, so that the surface of any nearby circulating platelets that just happen to be taking a stroll in the vicinity, they become sticky. And so they stick on to the first layer of the aggregated platelets that are already there. And they release more ADP. And then more platelets come by and stick on. And so the whole thing builds up like that as a plug. And of course the second step of hemostasis is blood clotting. So what that does is it reinforces the platelet plug. It converts the blood in the area of the injury into a non-flowing gel. So clotting factors here are always present in the blood, and they're always in an inactive form. When a vessel becomes damaged, the collagen is exposed, and that triggers the cascade of reactions that lead to a successive activation of clotting factors, which result in the conversion of fibrinogen to fibrin. So I'm going to talk now about the blood clotting cascade, not in detail at all, barely touching the iceberg, as one says. But the thing about the blood clotting cascade is that it's a series of steps, and there are 12 clotting factors involved. Calcium ions are needed at different points to keep it activated, to keep it moving, keep it ticking over, and that occurs throughout the cycle. But it all ends up, the cascade all ends up in a final common pathway whereby factor X is activated, a substance called prothrombin is converted to thrombin, and fibrinogen is cleaved to form fibrin. That's the main point. So what else do we know about clotting? About clot prevention? Is that possible? Is that feasible? How does that occur? Well, it happens through tissue plasminogen activator. And what that does is it converts plasminogen to plasmin, and so prevents inappropriate clots forming when you don't need them to be there. So it's used clinically as a clot buster. Then clot dissolution. This what happens to clot when you're done healing? What do you need? How do you need to get rid of it? Well, what happens is that plasmin comes along and dissolves the clot. So plasmin is made from plasminogen via various clotting factors, and so phagocytic white blood cells then come along and remove all of the end products. So another thing to keep in mind here is a little substance called thrombomodulin, and this binds thrombin so that no fibrinogen conversion can take place, because we don't want that to happen. And it also, thrombomodulin also activates protein C, which is an anticoagulant, which is also very important. When abnormal blood clotting occurs, you get a thing called thromboembolism. So a thrombus is a clot that attaches to a vessel wall, and an embolus or an emboli, they are free floating clots, which are really nasty, cause a lot of damage. And the thing with them is that there's lots of different factors that can increase the risk of getting thromboembolisms, including atherosclerosis, slow-moving blood, an imbalance in the clotting or anti-clotting systems. And then another thing about abnormal blood clotting is you can get haemophilia, like the Russian Tsars in Europe. And haemophilia is basically excessive bleeding due to the absence of a clotting factor, or a very important factor in the clotting cascade, of which there are very many. So haemophilia A is when factor 8 is missing, and haemophilia B is when factor 11 is missing. And that's the story of the blood.